The blueprint of all life is the DNA molecule. It's like a tiny zipper, wound into a corkscrew or helical shape. Only it's 6 million times smaller and packed into the nucleus of cells, where many thousands of these DNA molecules together form structures called chromosomes, which are vast libraries of blueprints for the construction, regulation, and duplication of the basic unit of life, the cell. The DNA molecule, when untwisted, looks something like this, with rails of alternating sugar and phosphoric acid molecules. The rungs of the ladder are the keys to the DNA blueprints. Each rung consists of a purine molecule combined with a pyrimidine molecule. DNA contains only 2 kinds of purine, adenine and guanine, which combine with only 2 kinds of pyrimidine, thymine and cytosine. These 4 molecules, represented by their first letters, can combine in only 4 ways. The order of these pairs along the DNA molecule is the genetic code, which forms part of the blueprint for the manufacture of a particular protein. But how do a series of chemical rungs on a ladder actually influence the construction of a protein? As with any code, like the magnetic information coding music on a tape, some means is necessary to read the code. If a sound system or a cell is to make use of it. Imagine if the designers who dream up a modern supersonic aircraft with its thousands of blueprints throw them all away after the prototype is built. Without copies of the blueprints, no more identical aircraft could be built. If the DNA code cannot be read in order to make copies for future generations, any accident that creates life on Earth could not be passed on. The precious master audio tape is first decoded in order to copy it into another. We now explore how DNA code is first decoded to make a copy of the DNA blueprint. For most organisms, life begins with a fertilized egg. Each sex cell, both a male, or a sperm cell, and a female, or an egg cell, contains only half the DNA molecules of other cells. When the egg cell is fertilized, the DNA molecules from the father and mother together form the full genetic complement passed on to the offspring. Now cell division, or mitosis, begins. In order for an organism to develop, each DNA molecule must be duplicated precisely and passed on to each new cell, until finally a fully formed, multi-celled individual plant or animal is formed. What's new is a process of DNA decoding and copying, which has taken place 10,000 million times. Except for occasional accidents, every molecule of DNA is an exact copy of a DNA molecule from the original fertilized cell. A remarkable copy job. Just how is it accomplished? The key to this copy process is the architecture of the DNA molecule. The spiraling rails of sugar, alternating with phosphate, leave room for only 2 kinds of rungs. Either a purine linked to a pyrimidine, or the reverse, a pyrimidine linked with a purine. There is too much room for one pyrimidine linked to another, and too little for a purine linked to another purine. But the conditions are even more limited when we look at the specific kinds of purine and pyrimidine. Purines and pyrimidines combine through a link-up between nitrogen and oxygen, through an intermediary hydrogen atom. There are other possibilities. In each case, one of the bonds is strong and the other weak. Adenine and thymine are capable of forming two of these bonds, which restricts them in DNA to combining only with each other. In contrast, both guanine and cytosine form three of these bonds, so they too must always pair up. Just what has all this got to do with decoding and replicating the DNA molecule? The strict pairing limitation, or complementarity, is central to the process of accurate DNA replication. When a cell undergoes mitosis, the DNA double helix unzips like a zipper and unwinds with the help of a special enzyme which decouples the hydrogen bonds between the purine and the pyrimidine. The result is two long strands of nucleotides, each consisting of a sugar phosphate rail section, together with a unit of adenine, thymine, guanine, or cytosine. Now, besides these unzipped DNA molecules, there is a large selection of free-floating nucleotides in the nucleus, identical to sections of DNA strands. At this point, complementarity comes into play. An adenine base on the unzipped DNA strand bonds with a free-floating thymine nucleotide in the nucleus. A guanine pairs with a cytosine. As each acquired nucleotide takes up its position, the phosphate of one nucleotide bonds to the sugar of the adjacent nucleotide to reproduce the characteristic sugar phosphate rail of the DNA molecule. The left-hand rail of this DNA molecule rebuilds a right-hand rail that exactly matches the original. And in the same way, the original right-hand rail rebuilds its left-hand complement. As the DNA strands rebuild, they twist into the characteristic double helix, two identical molecules where once there was one. Ladder-like, corkscrew-like, zipper-like, the two new DNA molecules are ready for distribution, one to each of the two daughter cells which are formed by cell division. Each daughter cell has inherited the original genetic information found in the original cell. In the process as well, two prime characteristics of life have been fulfilled. That of reproduction and that of growth. The continuity of the individual cell is ensured, and with it, the continuity of the organism as a whole.